Now, of course, mRNA rates can vary substantially between individual mRNAs, and this is just an example of this, looking at 3 different yeast mRNAs. Here at the 0 time, we block transcription of these mRNAs, and you can see that some, like the PGK message, last a long time. Others, like the MFA2 message, are extremely unstable and degrade quickly. In mammalian cells, the average rate of decay of mRNAs is about 8 hours, but again, those can vary tremendously. For example, the C-Phos mRNA has a half-life of about 15 minutes and degrades very quickly, whereas the beta-globin mRNA, which lasts for about 17 hours of a half-life. So there's tremendous differences in these decay rates between individual messages. One feature of mRNAs that controls their decay rates are what people have referred to as so-called stability elements. These are short sequence elements within mRNAs that control the rates of RNA turnover. So for example, within the C-Phos mRNA, there's an AU-rich element, or a so-called ARE, which can be transferred to the globin mRNA and then cause a rapid deadenylation and rapid decay after deadenylation. And this contrasts to the globin message, which is normally slow for those 2 steps. So these so-called stability elements then are part of the features of mRNAs which dictate their different rates of RNA turnover. Many of these stability elements serve as binding sites for proteins or microRNAs that then control RNA degradation. In this example I'll use here today, the PUF3 protein is a protein which binds to a short sequence element in the 3' ends of yeast mRNAs. And when it does so, it recruits the CCR4 deadenylase complex and subsequently can also increase the rate of decapping of that message. And that's a general principle that we see for many of these instability elements, is that they recruit these transacting factors that then directly or indirectly promote the degradation of the message. Now, one of the striking features of RNA decay and translation is kind of how they're intertwined with each other. And I want to highlight this by pointing out the distinct roles of the caps in both translation and RNA degradation. In translation, the cap and the poly A tail serve as binding sites for RNA binding proteins, which then assemble a complex which promotes ribosome loading. Conversely, during degradation, these are the 2 structures the main nucleases in the cell target. The poly A tail is removed by the CCR4 or PAN2 complex, ultimately leading to decapping of the message by the decapping enzyme. And once the message is decapped, as far as we know, the 5' to 3' exonuclease degrades the message extremely rapidly. So in general then, because of these 2 roles, translation and mRNA decapping are inversely related. So during translation, you have a complex which promotes ribosome loading. During degradation, you lose that complex and you promote degradation. And consistent with that, various observations support this kind of inverse relationship. If you alter the rate of translation initiation by mutations or alterations to the RNA, you increase the rate of decapping. Similarly, many sequence elements which people identified as either regulating translation or regulating decay do both. For example, the AREs I've talked about, which promote mRNA degradation, often under slightly different conditions, will also repress translation. The way we've interpreted this in general is that there are really 2 different types of RNA protein complexes existing in the cell. And what I'll call the translation mRNP and the decapping mRNP really are distinct. And for example, here will be one type of the translation RNP, and that it would have to then disassemble this translation initiation complex to form an alternative complex, in this case with the decapping complex, in order to be degraded. And the kind of evidence that supports this model comes from either co-immunoprecipitation experiments where RNAs that are bound by translation factors are not bound by decapping factors and vice versa. And also cell biological experiments which indicate that the mRNA decapping factors are not associated with polysomes. Polysomes are the ribosome mRNA complexes where proteins are being produced, but instead accumulate in these discrete cytoplasmic foci referred to as P-bodies. And these P-bodies, as I mentioned earlier, are found throughout eukaryotic cells. This is just an example in mammalian cells. And they tend to contain a core set of proteins, including the decapping enzyme, various proteins involved in degradation of those, in activation of decapping, as well as the exonuclease, which degrades following decapping exon 1.